Proteins are the building blocks of life. They carry out countless tasks in our cells. Proteins rarely act alone. They interact with each other to form intricate networks. These interactions are essential for life. Understanding them is key to unlocking the secrets of cellular function. Proteins interact with each other in various ways. These interactions can be strong or weak, permanent or temporary. The type of interaction influences the function of the proteins involved. Studying protein interactions provides valuable insights into how cells work. These interactions are vital for many cellular processes. They are involved in everything from cell signaling to DNA replication. By understanding these interactions, we can develop new drugs and therapies for diseases. Some proteins form stable complexes with multiple subunits. These complexes act as molecular machines. Hemoglobin, the protein that carries oxygen in our blood, is a classic example. Hemoglobin consists of 4 protein subunits bound together. Each subunit binds to an oxygen molecule. This allows hemoglobin to efficiently transport oxygen throughout the body. The 4 subunits work together to enhance oxygen binding and release. Another example is the pyruvate dehydrogenase complex. This complex is involved in energy production. It helps convert pyruvate, a product of sugar breakdown, into acetyl-CoA. Acetyl-CoA then enters the citric acid cycle to generate energy. The pyruvate dehydrogenase complex is a huge molecular machine. It consists of multiple copies of 3 different enzymes. These enzymes work together in a coordinated manner. This allows for efficient conversion of pyruvate to acetyl-CoA. Protein interactions can also be dynamic. This means they form and break down rapidly. These interactions are crucial for processes that require quick responses. Examples include cell signaling and immune responses. Dynamic interactions involve weak bonds between proteins. These bonds allow for rapid association and dissociation. This flexibility enables cells to adapt to changing environments. One example of a domain involved in dynamic interactions is the SH3 domain. The SH3 domain is a small protein module found in many signaling proteins. It binds to short, specific sequences in other proteins. These interactions mediate signal transduction pathways. They control various cellular processes. Scientists use various techniques to study protein interactions. One powerful technique is the yeast-2 hybrid assay. This method uses yeast cells to detect protein-protein interactions. In this assay, 2 proteins of interest are fused to different domains of a transcription factor. If the proteins interact, they bring the transcription factor domains together. This activates the transcription factor. The activated transcription factor then turns on a reporter gene. By observing the reporter gene's activity, scientists can determine if the 2 proteins interact. The yeast-2 hybrid assay has been instrumental in identifying novel protein interactions. It has helped uncover new drug targets. Understanding protein interactions is crucial for understanding how cells function. Dysregulated protein interactions can lead to diseases. By studying these interactions, we can gain insights into disease mechanisms. This knowledge can lead to the development of new therapies.